Hello and welcome everyone to this podcast for genetics. In the last podcast, we talked a lot about the history of heredity and began to introduce Gregor Mendel. And now in this podcast, what I want to focus on are the crosses that Gregor Mendel did. And we're going to begin with, in this podcast, talking about monohybrid crosses. In talking about the monohybrid crosses, we're going to talk about what are known as his 4 conclusions and his 1 law of segregation. In the next podcast, we'll talk about his law of independent assortment. So first, let's begin with what a monohybrid cross is. A monohybrid cross is a cross between parents that differ in only one trait. In this podcast, we're going to focus primarily on one of the traits that Mendel looked at in his plants, and that would be the trait of round versus wrinkled seeds. So in a monohybrid cross, one of the parents, in this example anyhow, one of the parents would have round seeds and the other parent would have wrinkled seeds. You could also have a monohybrid cross where they both are round or they are both wrinkled. So let's look at Mendel's first cross. And we're going to talk about pea plants that had round seeds and pea plants that had wrinkled seeds. And what Mendel knew, as well as anybody else in this day, they would have known if you took 2 pure breeding round plants or took 2 pure breeding wrinkled plants, pea plants, you would get all round, or you would get all wrinkled with the second cross here. Now, as I just described this, I mentioned the term pure breeding. A pure breeding plant is just as I just described. When you take one plant that has a certain characteristic and you cross it with another plant that has that same characteristic, and all you get is the same phenotype. So in this case, round times round, all round, wrinkle times wrinkle, all wrinkled. So it was important that Mendel began with these pure breeding strains. That way he knew there was nothing else in the genetic background that could confuse his crosses. So anytime you hear me use the word pure breeding, you can assume that when you cross it with itself, all you're going to get is the same progeny. Pure breeding strains are homozygous for a trait. They could be homozygous dominant, like big A, or they could be homozygous recessive, like little a. So let's begin by talking about one of Mendel's first crosses. He took a pure breeding round pea plant and crossed it with a pure breeding wrinkle-seeded plant. When this cross was initially done, they wouldn't have known exactly what they could have gotten. They might have gotten all round. They might have gotten all wrinkled. They might have gotten both. Or they might have gotten a blend where the pea plant was not perfectly round, but not perfectly wrinkled. Now that's harder to draw, but we'll draw something like that. Now remember, when you do an experiment, you don't know the results, because that's why you're doing the experiment. So when they did this cross, what they got was all round. But we now know why you get all round. You get all round because this is big R, little r, little r. And when you cross these together, you get all big R, little r. And we'll break this down in just a moment so you can see how this happens. But I just want to stress a couple points here. One is that Mendel didn't know anything about genes or alleles or chromosomes. He was just doing these crosses and observing and analyzing the results. So I want to break down this cross in a little bit more detail and use some terms we haven't used yet. Whenever a cross is done, we start with the P generation, which is simply the parental generation. And in this case, remember, we took a round plant and we crossed it with a wrinkled plant. And the results were all round. This generation, we always call F1. F1 means first filial generation. I suspect gardeners of the day knew if you did this cross, you would get all round. But remember, Mendel was a meticulous kind of guy. And so he did this next generation because he was curious what would happen if you crossed these 2 plants together. And so what he did was he would take 2 of these round-seeded pea plants and cross them with each other. And what he got was a mixture of round and wrinkle-seeded plants. I also suspect the gardeners of the day also knew that you would get this result. But I think it's probably safe to assume that Mendel was the first who decided to count them. And what he discovered was not that there was more of them that were round, but approximately 3/4 of them were round. In fact, in his first experiment, what he discovered was that 74.7% were round and 25.3% were wrinkled. Now later on, what we're going to do is something called a chi-square. So I'm just going to advertise it here. In fact, we'll do it for the next class. We use a chi-squared to determine if 74.7% and 25.3% is statistically equivalent to 3 to 1, or 75% to 25%. Turns out, in this case, it is. But we'll go through this fabulous test that you'll grow to love. We'll do that in class next time. And I just remembered, I forgot to label this generation. This generation is known as the F2, the second filial generation. After doing this first set of crosses, Mendel came up with his 4 conclusions and ultimately the law of segregation. The first conclusion he came up with is that the F1 plants, these here, must inherit genetic factors from both parents, even if they don't express that trait. So what he's saying here is that these pea plants had to inherit genetic information from this parent and this parent. It's easy to understand this parent because they're all round. It's more difficult to understand this parent because none of these are wrinkled. But he believed that they must have inherited some genetic factor from this parent because, lo and behold, you pick up wrinkled plants down here again. With this conclusion, he also believed that each parent must have 2 copies of each genetic factor. And this is the last time I'm going to call it genetic factor, because we now know what was considered a genetic factor then is really an allele. I should also point out that he was very confident in making these claims. He did greater than 60 crosses, and he also did reciprocal crosses to see that he got the same results. A reciprocal cross would be something on this lines, as if this one here, the rounded plant, was male, and this one here was female. Moreover, if this one was the female, and this one was the male, he'd still get similar numbers, suggesting that the sex of the plant did not have anything to do with what he was observing. That what he was observing was just a general phenomenon. We now can look back and say he got lucky with the traits he looked at, because if any of these traits were on an X chromosome or sex-linked chromosome, he might not have gotten these same results. So based on what we know now, we can go ahead and say this is an RR, a little r, a big R, little r, big R, little r, big R, little r, big R, little r. And so he took 2 of these, so they were big R, little r, big R, little r. And we now know, and we'll talk more about this when we talk about a Punnett Square, but we now know that these progeny down here, 1/4 of them were big R, a half of them were big R, little r, and this last quarter was little r, little r. How these combinations appear will become evident in a little bit. Okay, let's talk about Mendel's second conclusion now. These alleles separate from each other during gamete production. This happens so each gamete only gets 1 allele. It's important to note that Mendel's conclusions were made without knowledge of mitosis or meiosis, chromosomes, or that DNA was the genetic material, which I always find to be very impressive, that he was able to reason between these simple P crosses that there had to be 2 alleles, and that when these plants then make gametes, that each gamete would get 1. Same thing here, each gamete gets 1. So like here, each of these plants will make 2 gametes, big R in one of the gametes, little r in the other. Same thing here. And when these combine, they give these progeny. But he recognized that they had to separate, and that they had to end up in each of these gametes. His third conclusion had to do with his concept of dominance. In general, what he meant by this was that when 2 alleles are present, one of them will be seen as a phenotype. So in this case here, we have 2 alleles, big R and little r. Big R is dominant to little r, and little r is recessive to big R, such that big R, big R, big R, little r, big R, plants will be round. Rather, their seeds will be round. And only in this combination, little r, will the seeds be wrinkled. So his concept of dominance simply states that of 2 alleles, one will always be dominant over the other. Later, we're going to learn that this isn't always true, but for right now, let's just consider that one is always dominant to the other. Okay, next let's talk about Mendel's fourth conclusion. When alleles separate from each other, they do so with equal probabilities. When this plant here makes gametes, half of them are going to be big R and half of them will be little r. We don't see that more of them are big R or more of them are little r. Sometimes one might confuse the idea of dominance with the idea that more of the gametes will have the dominant genotype, but that's not the case. Dominance only refers to the phenotype of the plant. Gametes are made, and when they are made, the alleles are segregated with equal probability. These 4 conclusions he was able to make because it's the only way the math works out. When he counted the p's, these were the only possible conclusions he could make. Turns out he was right. It's pretty impressive. These 4 conclusions then led to ultimately Mendels First Law, which is also known as the principle of segregation. This principle of segregation is going to sound very familiar to the 4 conclusions because it's built from it. And that is that each organism contains 2 alleles for each trait. Alleles segregate so that each gamete gets one or the other allele. Allele segregation is random and equal. When these alleles segregate, we know half will get the big R, half will get the little r. Which gamete gets which is completely random. That part is the random, but it's equal in that you should get 50% of each. Mendel's first law deals only with one allele pair. His second law will deal with more than one. That's the main difference. As a bit of a spoiler alert for the next podcast. Don't tell anyone. Okay, I know I've said this before, once or twice, and I'll probably say it again. But the impressive thing about Mendel's experiments is that he was able to do them, and he was able to make these conclusions without that fundamental knowledge of meiosis or chromosomes. However, once we understood meiosis and the chromosome, it was easy to see how meiosis explains Mendel's conclusions. So in showing how meiosis explains Mendel's conclusions, let's consider his F1 cross, where he took a big R, little r, round-seeded plant, and he crossed it with a big R, little r, round-seeded plant. Since these are identical, I'm only going to talk about meiosis with one of them. So here's what the cell looks like. In G1, we have these 2 chromosomes where we have big R and little r. It goes through S phase, and it's sitting here at G2. And because it went through S phase, remember, we have 2 copies of each chromosome. It then goes into meiosis. It goes through prophase, and I'm going to draw metaphase next. So meiosis 1, it enters. And at meiosis 1, remember, it looks something like this, with the metaphase plate coming down here, the homologous chromosomes on either side of the metaphase plate, something like that. So this was metaphase 1. It then completes metaphase 1, anaphase 1, and telophase 1, and it'll produce these 2 cells, where one of them contains both copies of R, and the other contains both copies of little r. We're also assuming at this point no crossing over is happening. We're just keeping this simple. I should, right here, just to keep it legal in case you're keeping score at home, meiosis 1 completed. All right, each of these cells now can enter meiosis 2, and what I'm going to draw here in meiosis 2 is metaphase 2. We have our metaphase plates, and now remember, this should be all old information for you, but we're separating out the sister chromatids in both cases. Big R, and then little r, little r. These then go through anaphase 2 and telophase 2, in which time this chromosome gets into a gamete, this chromosome gets into another gamete, and same here. So we end up with 4 cells. Two with big R, 2 with little r. Again, this supports Mendel's conclusions. We have 2 alleles. Through the process of gamete production, the alleles separate from each other, such that the gametes contain either big R or little r, and we have equal probability of having a big R or a little r gamete. Now remember, I only showed you this for one plant, but the exact same thing is happening in the other plant. So this plant is making these 4 gametes per round of meiosis, and so are these. Let's consider these 4 gametes in setting up what is known as a Punnett Square, which perhaps you've already seen this in an earlier class or from high school. I'm going to keep these written down here for just a moment because I want to talk about this thing called a Punnett Square. When we talk about Punnett Squares, often we'll write them something like this, where we're taking big R, little r, times a big R, little r. And then we draw this grid like so, and we place a big R here, and a little r, big R here, and a little r. And then we combine them. But I don't want to do that. I want to do this in a slightly different way. Because what's on these axes aren't just random letters, they're gametes. So let's draw them as such. And remember, these gametes came from meiosis, just like we see here. So one of the parents shown here is big R, and the other parent is little r. Same thing here. When these gametes come together, they'll form an organism. I'm going to show the organism as a square, just to make it distinct from the circles that I'm showing as gametes. These organisms that result, or maybe I should call them progeny, the progeny of this cross, will contain cells that have each of these chromosomes, one from one parent, one from the other. This parent here, for this progeny here, contributes the big R gene. And to this progeny, this parent contributes a big R gene. And down here, this progeny gets a big R from this parent, and it gets a little r from this parent. Big R from this parent for this progeny, little r from this parent here. And then both parents for this last progeny down here provide the chromosome with the little r. I wrote it this way because I want you to remember that every time we jot down a Punnett square to help us do a problem, what we're really jotting down, or what we should really be thinking about, is that it's the chromosomes that we're following. And as we move forward and start talking about how this allele on this chromosome is linked to other genes on this chromosome, it'll make that discussion a lot easier if we don't forget that we're talking about chromosomes. Okay, having done this Punnett square, we can make certain kinds of predictions now. We can make a phenotypic prediction. From this, we would predict this progeny, and this progeny would have the dominant phenotype of round seeds. So we would say 3 round seeds to just this one down here, which should be 1 wrinkled. So our prediction for the phenotype of this generation here is 3 round seeds to 1 wrinkled seeded plant. We can also make a prediction about the genotype. And again, remember, these are just predictions, but we would predict the following genotypes. We'd expect 1 RR to 2 big R little r's, these two here, to 1 little r little r. Once you've made your predictions, as I've said, you then do the cross. A podcast coming up, we're going to figure out how we take this prediction and compare it to the actual cross results. I said it before, I'm going to say it again, it's the chi-square analysis. We expect 3 to 1 phenotypic, but is that what we get? The chi-square analysis will help us determine that. Now for simplicity, I've only focused on the pea plants that Mendel worked with. And I only focused on this one characteristic of seed shape, round versus wrinkled. But everything we've talked about in this podcast, we could say the same for another trait found in pea plants, or any monohybrid cross found in other plants or animals. Okay, this is the last thing I want to talk about in this podcast. And it's centered around looking at a dominant phenotype. And we might as well stick with our very good friend here, the rounded pea seed. If somebody gave you a plant and it had round pea seeds in it, how would you know that this plant was big R, homozygous dominant, or big R, little r, the heterozygote? You have no way of knowing. Both of these will give you the dominant rounded pea plant. So to confirm either big R, or big R, little r, we do what is called a test cross. A test cross strain, so test cross strain, is always homozygous recessive. So in this case, it's going to be little r, little r. Let's go back to our problem here. We're given this pea plant, and we don't know if it's big R, or if it's big R, little r. And so what we're going to do is try to make a prediction here and do a Punnett square. And so we're going to take the plant that our friend gave us, and we're going to test cross it to a homozygous recessive plant. So we're going to test cross it with a little r, little r. So I'm going to write TC here, so we remember it's a test cross strain down here. And up here, this is our plant of unknown genotype. By doing a test cross, we can figure out which one it is. Now, if the genotype of the plant your friend gave you is big R, and you do a test cross, then all the progeny should be round, have round seeds, that is. Let's go and draw this out here. This one would be big R, little r, because of these two. This one would also be big R, little r, from these two. This other one would be big R, little r, and so would this one, from these two or these two. So, the plant they gave you had the genotype of big R, and you did a test cross, this is all you would see, are round plants. Now, if the plant they gave you was big R, little r, still rounded plants because of the dominance of R over little r, what would the results be? If you want, you can go ahead and hit pause, and then come back here, and then you can see the results, just for practice. Okay, so you would predict the following progeny if this was a heterozygote. You would still get 2 big R, little r's, shown here, but these two here would be little r, like that. This little r combining with that little r, and this little r from the test cross plant with this little r. So you get 50% round and 50% wrinkled. we will use test cross every now and then as we move through these next couple of weeks. So anytime you hear the term test cross strain, you should immediately think of a homozygous recessive strain. You should also immediately think, I'm going to use a test cross strain to help determine the genotype of a dominant phenotype. All right, well, that's all I have for this podcast. If you have any questions at all, please make sure you come see me. If not, I will see you in class. Bye.